Welcome to the Deep Dive, the show that cuts through the noise to get you well-informed fast. Have you ever really stopped to consider what it would take to read every single scientific paper published on, a really complex topic?
It's mind-boggling.
Right. Or maybe imagine trying to analyze the entire human genome letter by agonizing letter. It's basically an impossible task for any one person.
Absolutely.
The sheer, just overwhelming volume of biological data we have today it's staggering. It's been a challenge pushing the boundaries of discovery for decades, kind of constantly reminding us of our human limits.
Yeah, it really puts things in perspective.
But here's where things get really interesting, really transformative. Today, we're diving into a world where that impossibility is, it's becoming a tangible reality. We're embarking on a deep dive into how artificial intelligence, especially things like generative AI, and advanced machine learning isn't just assisting anymore, it's fundamentally revolutionizing the life sciences.
It really is a revolution. We're talking about truly groundbreaking stuff.
Exactly. Yeah. Things like human medical genomics, proteomics, data analysis, synthetic biology, drug discovery, even, how we monitor our planet's delicate ecosystems. It's touching everything.
It's incredibly broad.
So our mission for you today is basically to give you a curated shortcut, a way to understand this field that's evolving so rapidly and having such a deep impact.
Yeah, trying to make sense of it all.
We'll unpack the how and the why AI is making these transformations. From diagnosing those really rare genetic diseases to designing entirely new proteins from scratch, and even optimizing complex industrial processes.
And crucially, we can't ignore the challenges.
Absolutely not. We'll also explore the significant hurdles and the really profound ethical questions that pop up when you start deploying technology this powerful responsibly. So get ready for some, hopefully some insightful moments, maybe even some moments.
Definitely. And for this deep dive, we've pulled together a pretty rich stack of recent research and reviews.
Good stuff.
Yeah, I think so. We've got systematic analyses on generative AI in human medical genomics, some really in-depth studies looking at AI's role in proteomics data analysis, and also broader perspectives covering AI applications across, various life science domains.
Okay.
So the plan is to extract the most important nuggets of knowledge, maybe some surprising facts, just to give you a really comprehensive and hopefully engaging picture of where things stand right now.
All right. Sounds like a plan. Let's unpack this then. When we talk about AI in biology, what exactly are we talking about? Let's maybe start with a basic understanding. What is AI and how did we get here?
Good place to start. So at its core, AI is really the science and engineering behind creating intelligent machines.
Right. Machines that think.
Sort of. Well, machines capable of tasks that usually need human intelligence. Things like learning, reasoning, making decisions, solving problems. Fundamentally, it's about looking at huge amounts of data, finding patterns, and then making predictions based on those patterns.
And this isn't brand new thinking, is it?
Not at all. What's fascinating is how far back these ideas go. If you connect this to the bigger picture, the roots are way back with Alan Turing in 1950.
The Turing test guy.
Exactly. He was pondering self-learning machines, came up with the famous Turing test, a benchmark for machine intelligence. That was truly radical thinking back then.
And then we had things like Deep Blue, the chess computer.
Right. Fast forward to 97, IBM's Deep Blue beats Garry Kasparov. Huge moment. But Deep Blue is mostly what we'd call symbolic AI.
Meaning?
I mean, it operated on a meticulously predefined set of rules. If this, then that, basically. The big shift we see today is that modern AI can kind of learn on the fly from experience. It moves way beyond those rigid preset rules. It's all about adaptive intelligence now.
Okay, so that learning on the fly, that's machine learning, ML, and deep learning, DL, right? They're the engines driving this.
Precisely. Those are the key technologies. ML, as a core branch of AI, lets systems learn and get better from experience without someone explicitly programming every single step.
Like teaching by example.
Exactly. Think of giving a student a huge stack of examples and letting them figure out the underlying rules themselves. That's the essence of it.
And within ML, there are different approaches.
Right. Two primary ones are really relevant in biology. First, you've got supervised learning. This is what you use for classification tasks.
Okay.
Here, the models learn from labeled training data where you already know the answer, so you train it to predict specific outputs.
Like telling diseased tissue from healthy tissue.
Exactly. Or predicting high versus low protein expression. You show it labeled examples, and it learns to make predictions on new, unlabeled data.
Got it. And the other type?
That's unsupervised learning. This is more for exploratory data analysis. It's about identifying hidden patterns in data without any pre-existing labels.
So finding structure in the chaos.
Pretty much. For instance, you might use it to cluster proteins based on similarities in their expression profiles. The model discovers these natural groupings on its own, potentially revealing relationships you didn't even know existed.
Interesting. And when the patterns get really, really complex, that's where deep learning steps in.
That's right. Deep learning, or DL, is a subset of ML. It uses these things called artificial neural networks, but with multiple deep layers.
Layers upon layers.
Exactly. And these layers let it model and understand incredibly intricate, often nonlinear relationships in the data. It's truly remarkable what these deep networks can pull out, especially with the kind of massive, complex biological data we're generating now.
So within deep learning, are there specific types of networks, specific architectures that are particularly important in biology?
Yeah, definitely. We see a few key ones pop up again and again. There are convolutional neural networks, or CNNs. They're primarily used for analyzing visual imagery, think complex tissue scans, and also sequence data, like protein sequences.
Okay, CNNs for images and sequences. What else?
Then you have recurrent neural networks, RNNs. These are specifically designed to recognize patterns in sequences of data where order matters. So think time series proteomics data, or maybe long stretches of genetic code.
Makes sense. Sequences over time or space.
And finally, we should mention autoencoders. These are often used for dimensionality reduction.
Meaning simplifying complex data.
Exactly. They help distill the essential features from incredibly complex data sets while getting rid of some of the noise. This really helps with visualizing the data and interpreting what's going on.
Okay, CNNs, RNNs, autoencoders. Got it. Now, you mentioned generative AI earlier. That feels like a big step change.
It really is, because unlike traditional AI that mostly analyzes or classifies existing information,
Generative AI creates new stuff.
precisely, it has this unique capability to generate entirely new data. It's not just recognizing the cat in the picture, it might generate a completely novel image of a cat that's never existed before.
Wow. And the engine behind a lot of this modern AI, especially language stuff, is this transformer architecture.
That's right. Transformer architectures are really the state-of-the-art backbone for many of these models. They're known for their exceptional ability to understand context in unstructured data, particularly text.
And within transformers, there are different types, like BERT and GPT. We hear those names a lot.
Exactly. Those are the two big ones. So BERT, that stands for bidirectional encoder representations from transformers, is an encoder-only model.
Encoder only.
Yeah, meaning it's primarily designed to understand input text. And it does it bidirectionally, looking at context from both left to right and right to left at the same time.
Ah, so it gets the full picture.
Right. This gives it a much more complete understanding of the context, making it really effective for tasks like classifying text or pulling out specific named entities like gene names or disease names from scientific papers.
Okay, so BERT understands. What about GPT?
GPT generative pre-trained transformer is the other side of the coin. It's a decoder-only model.
Decoder. So it generates.
Exactly. It excels at generating text. It predicts the next word based on the words that came before it, kind of in a unidirectional flow. Think text generation, complex translation, summarizing dense scientific articles. It's about creating new, coherent, contextually relevant content.
Makes sense. BERT reads. GPT writes.
Yeah.
Oversimplified, I know.
A useful shorthand. And beyond those, we also have generative adversarial networks, or GANs.
GANs, right? Those sound adversarial.
They kind of are. It's a specialized setup for data generation. You essentially have two neural networks competing. One network, the generator, creates fake data, say, images or genetic sequences. The other network, the discriminator, tries to tell if the data is real or fake.
So they push each other to get better.
Exactly. This adversarial process forces the generator to produce incredibly realistic outputs. We see GANs used for synthesizing realistic medical images, complex biomedical data, even artificial genetic sequences.
Okay, that's fascinating. And then there's one more term that seems to be everywhere lately, foundation models.
Ah, yes, foundation models. These have truly exploded in relevance, especially in the last year or two.
So what defines them? What makes them different?
They're defined as models trained on vast and really diverse data sets. Crucially, this data is often unlabeled, meaning the models learn in a self-supervised way. They have to infer the patterns and relationships themselves.
Learning without explicit labels.
Right. And because they're trained on such broad data, they can then be fine-tuned for a whole range of specialized downstream tasks using much less additional specific data.
So the core learning transfers.
That's the key insight. Their ability to transfer learned knowledge to new domains is what's rapidly accelerating advances in so many areas: research, healthcare, genomics. The numbers speak for themselves. Publications citing foundation models jumped dramatically in 2024. We're talking from fewer than 10 annually before 2023 to potentially hundreds now. It really highlights their impact.
Wow. Okay, that sets the stage nicely.
Huh.
Let's zoom in now on one of the most impactful areas you mentioned, human medical genomics. Specifically, how AI is revolutionizing diagnostics.
Right. This is a huge area. When you think about lab diagnostics for inherited diseases, there are really two major goals, and they're interconnected. First, you need to establish the correct diagnosis for the patient. What condition do they actually have? And second, you need to identify the exact genetic cause, the specific genetic variation that's responsible.
Finding the needle in the genomic haystack.
Pretty much. And the typical workflow in medical genetics is quite intricate, but you can break it down into three key stages.
All right, stage one.
Stage one is the pre-analytical stage. This is all about patient prep and data organization, gathering patient history, clinical info, assessing genetic predispositions, collecting the biological samples.
Get round work.
Exactly. And crucially, it also involves a lot of literature review and knowledge aggregation, pulling information from vast scientific databases and papers.
Okay, pre-analytical.
Then comes the analytical stage. This is the core diagnostic phase where the actual data processing and interpretation happens. For next-generation sequencing, NGS data, this means calling the genomic variants, annotating them.
Figuring out what the sequence differences mean.
Right. And this often requires painstaking manual curation, checking against external databases. Plus, this stage often integrates insights from other things too, like MRI or CT scans, histology slides, even patient photographs, trying to build a complete picture.
Makes sense. Multimodal data. And the final stage.
The post-analytical stage. This is where you integrate all those diagnostic results into clinical decisions. Subtyping the disease, maybe clustering patients with similar profiles, aggregating all the findings, making informed decisions, and finally, generating comprehensive reports for the clinicians.
Okay, pre-analytical, analytical, post-analytical. So where does AI fit in? Why is it needed here?
Well, the why becomes really clear when you realize that traditional statistical methods and even older machine learning techniques often really struggle with the complex, high-dimensional data that's typical in modern genetics.
There's too much data, too many variables.
Exactly. And that's precisely the challenge that these state-of-the-art deep learning models, especially those transformer-based architectures we talked about, are now starting to address really effectively.
Okay, let's break that down then. Starting with the pre-analytical stage. How is AI helping with that initial groundwork, the literature review, the data gathering?
Well, our sources show a lot of focus here, particularly on large language models, LLMs. They really excel at this.
How so?
They're incredibly good at extracting structured knowledge from just vast amounts of unstructured text, biomedical literature, clinical notes. Imagine automatically pulling out and standardizing information for things like the Human Phenotype Ontology, HPO.
That's the system for categorizing clinical features, right?
Exactly. Or doing named entity recognition, NER, to automatically identify critical gene-disease relationships or links between phenotypes and specific genetic variants.
It sounds like it would save a ton of time.
Absolutely. BERT-based architectures are often fine-tuned specifically for these tasks, dramatically speeding up what used to be a very manual, very laborious process.
So extracting information. What else in this stage?
Well, LLMs are also getting very good at supporting biomedical and clinical question answering.
Like a specialized search engine.
Sort of, but more powerful. It ranges from retrieving facts from the literature to doing really fine-grained evidence extraction, say, to help interpret a specific genetic variant. We're seeing domain-specific Q&A systems trained on huge data sets like PubMed.
PubMed is the big database of biomedical papers.
Right. And other systems are optimizing question answering for prioritizing which variants are most likely to be disease-causing using a technique called retrieval-augmented generation, or RAG.
That sounds interesting. How does that work?
It's clever. Instead of the AI just trying to recall an answer from its training data, it actively goes and looks up relevant information from a trusted knowledge source like a curated database or recent papers, and then it builds its answer based on that retrieved, verified information.
Ah, so it checks its facts before answering. That sounds much safer for clinical use.
It definitely improves reliability and accuracy. It's less likely to just make things up or hallucinate, which is a known issue with some LLMs.
Okay, so AI helps find and retrieve existing knowledge. Is it also helping create new knowledge?
Yes, and that's a really exciting development. Transformer-based models aren't just extracting what's already written. They're being used to generate new biomedical knowledge and hypotheses.
How does that work?
We're seeing models that create new structured knowledge, like graph-based models that map out complex relationships between drugs, diseases, and patient phenotypes, allowing for new kinds of reasoning.
Finding connections you wouldn't see otherwise.
Potentially. And some models can even generate vector-based hypotheses to predict, say, disease gene associations that aren't explicitly mentioned anywhere in the training data.
Wow. That's moving beyond summarizing to actual discovery.
It's a pivotal shift. It allows us to potentially go beyond what we already know and uncover entirely new biological connections or therapeutic avenues.
Okay, one more area in the pre-analytical stage, risk stratification. How's AI helping there?
Right. Here, transformer models and LLMs are used to evaluate a patient's risk for certain diseases. They can process clinical notes, family history information, sometimes even multimodal health data.
To predict future risk.
Exactly. Assessing an individual's genetic predisposition based on various inputs, or identifying patients across large populations who might be at higher risk and could benefit from earlier screening or preventative measures. It helps clinicians prioritize resources.
Okay, that covers the pre-analytical stage. Now let's dive into the analytical stage, the real core of data interpretation. This is where a lot of the AI action is.
Yeah, this phase gets a huge amount of attention in the research, arguably where some of the most intricate AI applications are emerging. Let's start with medical imaging analysis.
Right, you mentioned things like MRIs and CT scans.
Exactly. Vision transformers, VITs, and other transformer-based models are increasingly being used here. The key is linking visual features from those scans with genomic data.
Combining sight and sequence.
Precisely. We're seeing these multimodal frameworks emerge. They might link imaging, like an MRI, with genetic data to help detect structural abnormalities. For example, some models align gene expression patterns with MRI scans for really precise tumor segmentation.
So helping doctors see the tumor boundaries more clearly?
Exactly. Others combine histology, looking at tissue slides, with genomic and clinical features to predict cancer risk. There are even examples of hybrid CNN transformer models designed to segment brain tumors more effectively, often guided by input from expert radiologists.
Like an AI co-pilot for the radiologist.
That's a good way to think about it, helping them spot subtle anomalies with potentially greater accuracy and speed.
That's amazing.
Yeah.
Does he go further? Can AI look at an image and predict genetic information directly?
Yes, it seems so. Models are being developed that can predict specific mutations or even chromosomal abnormalities, karyotypes, directly from medical images, histology slides, cytogenetics images, even facial photographs in some cases.
Facial photos, really?
Yeah, there's a tool, for instance, that aims to enhance the diagnosis of rare genetic diseases by combining analysis of facial images with clinical text information. It's looking for those subtle dysmorphic features that can be indicative of certain syndromes.
Incredible. Finding genetic clues in visual data. Okay, what about analyzing the genetic sequence data itself? The analysis of variant effects.
Right, moving from the pictures to the code, so to speak. Deep learning models, including those encoder-only transformers like BERT, are now used to extract really deep structural information from DNA, RNA, and protein sequences.
Going beyond just reading the letters.
Way beyond. For example, models like Informer are predicting the effects of non-coding genomic variants, changes in DNA that don't directly alter proteins but can affect gene regulation. And crucially, Informer is optimized to consider long-range interactions within the genome, sometimes up to 100,000 base pairs away.
Wow, interactions that far apart. That sounds important.
It's critical. Gene regulation is complex, and things happening far away on the DNA strand can have major effects. Older models often miss these long-range interactions. We also see models like Dean and BERT, which is basically BERT adapted to understand DNA sequences contextually, helping predict important regulatory regions like promoters or splice sites.
So AI is not just identifying sequence changes, it's predicting what those changes do, their functional consequences.
Exactly. Transformers are predicting how genetic variants impact things like protein structure, how proteins interact with each other, how genes are regulated, and even how a patient might respond to a particular drug based on their genetics.
Can you give an example?
Sure. Alpha missense is a well-known model that achieves state-of-the-art performance in classifying the pathogenicity of missense variants, those single-letter changes in DNA that result in a different amino acid in the protein.
So predicting if a variant is likely harmful or benign.
Right. Other models predict how mutations might alter protein interactions or even changes in DNA methylation, which is an important epigenetic marker in complex disorders like neuropsychiatric conditions. This lets researchers understand the potential biological impact of a genetic change with unprecedented detail before doing potentially laborious experiments.
And you mentioned bridging the gap between genotype and phenotype, connecting the genetic code to the actual disease or trait.
Yes, that's a huge goal. Transformer-based models are increasingly used to model these complex relationships. We see this in predicting polygenic risk scores, or PRS.
That's combining the effects of many small genetic variations to estimate overall risk.
Exactly. And genomic large language models are being developed to improve PRS prediction for common complex diseases like breast cancer, diabetes, and Alzheimer's. Other approaches are using multimodal data combining genetics, imaging, clinical info for predicting brain disorders, aiming for more personalized and mechanistic insights.
Okay, so we've analyzed the variants, predicted their effects. What about the final step in this analytical stage? Clinical variant interpretation, deciding which variants actually matter for this patient.
Right. This is where it all comes together for diagnosis. The goal isn't just discovery, it's determining which specific variants observed in a patient are truly causal or clinically relevant. And AI is helping here too. Tools like Genetic Transformer, or GeneIt, use fine-tuned LLMs to essentially emulate the reasoning process of an expert human geneticist. They try to identify the causative variants in rare genetic diseases by leveraging semantic understanding of the disease and phenotype-aware context.
So the AI is trying to think like an expert.
That's the idea. Trying to connect the patient's specific clinical features, phenotype, with the potential impact of their genetic variants, drawing on vast knowledge bases.
And this helps prioritize which variants to focus on.
Definitely. Some generative AI systems allow clinicians to interact with them in a user-friendly way, maybe asking questions in natural language to get summaries of the scientific literature related to a specific variant.
Yeah.
This helps contextualize things very quickly. Other models are applying attention mechanisms specifically to structural variants, larger genomic changes, to understand their impact on gene function.
So faster path from raw data to potential diagnosis.
That's the goal, reducing the time and effort needed for interpreting complex genomic data.
All right, that covers the analytical heavy lifting. Let's move to the post-analytical stage. This is where all the findings get synthesized into actionable clinical insights, right?
Exactly. You have all this output from the analytical stage, variant calls, interpretations, imaging analysis. Now you need to make sense of it for clinical use.
So how does AI contribute here?
One way is through patient clustering and subtyping. Transformer-based models can take all that data, genetic, clinical, imaging, and group patients with similar profiles.
Why is that useful?
It helps support differential diagnostics. Does this patient fit a known disease subtype? Or maybe they represent a new, previously unrecognized subtype. This can guide treatment decisions and prognosis.
Makes sense. Grouping similar patients. What else?
Data and results aggregation. AI is really good at fusing diverse data streams, taking variant data, integrating it with findings from the literature, maybe predicting patient outcomes based on similar cases, and even providing initial suggestions for treatment avenues based on the integrated evidence.
Handling that complexity again.
Precisely. It's computationally intensive, but incredibly valuable for getting a comprehensive view for patient care.
And the final output, the report.
Yes, clinical report generation and decision support. LLMs are increasingly being used to help generate clinical reports and summaries. Importantly, they can often provide explainable outputs, helping physicians understand the reasoning behind the AI's conclusions.
Explainability is key for trust.
Absolutely. These models also support clinical decision-making more broadly and can help automate certain clinical processes, all leveraging their improving semantic reasoning capabilities. Imagine an AI helping draft a detailed, accurate report about complex genetic findings, saving clinicians hours of manual writing.
That would be a huge help. Is there also a role for AI in just education? Training doctors?
Yes, absolutely. And this connects back to the bigger picture. There's a really promising role for generative AI in creating ethical and privacy-safe training materials.
How so? synthetic data again.
Exactly. For example, researchers have used AI, specifically GANs, using methods like StyleGAN to generate synthetic facial images of children with features characteristic of rare genetic conditions like Kabuki and Noonan syndromes.
To train doctors to recognize the signs.
Right. They used these AI-generated images to train pediatric residents, and the results were interesting. While real patient photos were still slightly more helpful overall, the synthetic images significantly increased diagnostic confidence and reduced uncertainty among the trainees.
So a safe way to practice recognizing rare conditions without using actual patient photos.
Precisely. It opens up powerful new ways to train medical professionals, especially for conditions they might not encounter frequently in person. There's also potential for using things like ChatGPT to generate patient information leaflets tailored to specific conditions or findings.
Fascinating. Okay, let's shift gears slightly. We've talked genomics. What about proteomics, the study of proteins? How is AI transforming that field?
Proteomics is another cornerstone of modern biology, absolutely. Studying proteins on a large scale gives us fundamental insights into how cells function, what goes wrong in disease, and where we might find new therapeutic targets. Proteins are the real workhorses of the cell.
But analyzing them is hard.
Extremely hard. It comes with some major analytical challenges. Proteomics data is inherently complex and just massive in volume.
What are the specific challenges?
Well, first, high dimensionality. You're often dealing with data sets measuring thousands of different proteins, and their levels are often interdependent. It's statistically very complex.
Okay, thousands of variables.
Then there's data variability. Protein expression isn't static. It fluctuates across different tissues, under different conditions, over time points. Discerning meaningful biological signals from this natural variation is tough.
Or trying to hear a whisper in a noisy room.
Exactly. And that leads to the third point. The data is often inherently noisy. Techniques like mass spectrometry or MS, which are workhorses in proteomics, generate data that contains background noise and often has missing values for certain proteins in certain samples.
Right, so the raw data isn't perfect.
Far from it. And finally, if you want the really deep insights, you need to integrate proteomics with other omics layers, genomics, transcriptomics, metabolomics. Data integration for that kind of holistic view is incredibly computationally intensive.
Okay, so complex, high-dimensional, variable, noisy data, hard to integrate.
Yeah.
Sounds like a job for AI.
Precisely. AI positions itself as a truly transformative tool here. It significantly enhances data processing, helps find those subtle patterns, improves predictions, and ultimately accelerates discovery, fostering a much deeper understanding of the proteome.
So what are some of the key applications? How is AI actually being used in proteomics?
Let's break it down. First, just identifying and quantifying the proteins themselves. Protein identification and quantification.
From that noisy MS data.
Exactly. Machine learning models, including deep learning, are used to process the raw mass spectrometry data. Algorithms like support vector machines or deep learning models analyze the peptide spectra, the fingerprints of protein fragments, to identify which proteins are present and how much of each there is. This improves both accuracy and speed compared to older methods.
And dealing with the noise.
AI helps there too. It's used to denoise the MS data, helping to distinguish the real protein signals from the background noise, which is crucial for getting reliable quantification.
Okay, so identifying and counting proteins, what about figuring out what they do?
That's functional annotation. AI models analyze protein sequence data and increasingly structural data too to infer a protein's function. Convolutional neural networks, CNNs, are particularly good at analyzing sequence information for functional clues.
Like predicting if it's an enzyme or a structural protein.
Exactly, or even identifying specific functional domains within a protein, the parts responsible for binding DNA, or interacting with other proteins, or catalyzing a reaction. It helps us understand their precise roles.
And protein interactions. Do proteins work alone?
Rarely. Understanding protein interactions, or PPIs, is critical. AI is used to predict these interactions by analyzing sequence data, structural information, and existing experimental evidence.
What kind of AI models?
Graph neural networks, GNNs, are showing a lot of promise here as they're naturally suited to modeling networks. Even natural language processing, NLP models are used, sometimes treating protein interactions like relationships in a text. The goal is to map out these complex social lives of proteins within the cell. Tools like the string database are valuable resources combining experimental data with computational predictions for PPI analysis.
Okay, interactions. Now, what about the shapes of proteins? Structure equals function, right?
Absolutely. And this is where AI has caused arguably the biggest revolution in biology in recent years. Structural proteomics.
You mean AlphaFold?
Exactly. The impact of models like DeepMind's AlphaFold has been nothing short of a game changer. It can predict the 3D structure of proteins from just their amino acid sequence with near experimental accuracy.
Which is huge for understanding function and designing drugs.
Absolutely critical. Knowing the precise 3D shape is fundamental for understanding how a protein works and how a drug might bind to it. And while AlphaFold is renowned for its accuracy, we also see other players like Meta's ESMFold. It might be slightly less precise in some cases, but its speed is incredible. It predicted structures for over 600 million proteins in just 2 weeks using a massive GPU cluster.
600 million. The scale is just, wow.
It's staggering. And beyond predicting the whole structure, AI also assists in things like epitope mapping.
What's that?
That's identifying the specific small regions on a protein surface, the epitopes, where antibodies bind. This is crucial for designing effective vaccines and antibody therapies.
Got it. And you mentioned the challenge of integrating different omics data types.
Right. Multi-omics data integration. This remains a major computational hurdle, but AI is making significant inroads. It helps effectively combine proteomics data with genomics, transcriptomics, metabolomics, and other data types to uncover those systems-level insights into cellular mechanisms. AI provides the tools to manage and analyze that complexity, giving us a much more holistic view than any single omic layer could alone.
That holistic view seems essential. Okay, let's zoom in on one really important application that spans across these omics: AI-driven biomarker discovery.
Yes, this deserves a dedicated focus. Biomarkers are incredibly important.
Remind us what they are.
They're basically measurable indicators of some biological state or condition. Think of blood glucose for diabetes or PSA for prostate cancer risk. They're crucial for diagnosing diseases, predicting prognosis, monitoring how well a treatment is working. Finding good biomarkers is a huge goal in medicine.
And finding them is hard.
Very hard, especially in complex diseases using high-dimensional omics data. That's where AI comes in.
So what's the typical workflow when using AI to find biomarkers?
It generally follows 3 key steps. Step 1, data collection and pre-processing. You gather the data, proteomics, genomics, whatever omics you're using from your biological sample.
Healthy versus diseased, for example.
Right. Then comes crucial pre-processing, data standardization, often called normalization, to remove technical variations or batch effects that aren't biologically relevant. And very importantly, feature selection.
Feature selection, meaning choosing what to focus on.
Exactly. Your raw omics data might have thousands or tens of thousands of features, proteins, genes, metabolites. Feature selection uses statistical or AI methods to narrow that down to a smaller set of features that are most likely to contain potential biomarkers, making the downstream modeling more efficient and interpretable.
Okay, collect, clean, select features. Step 2.
Step 2. AI model development. This is where you train AI models on your pre-processed labeled data, say samples labeled as healthy or diseased. The model learns the patterns associated with each group.
What kind of model?
It depends on the data and the goal. You might use supervised learning if you have predefined classes, like predicting disease status. Or you might use unsupervised learning if you're trying to discover completely new biomarkers or disease subtypes without prior labels.
Train the model. Step 3.
Step 3, validation and interpretation. This is critical. You have to rigorously validate the model's performance using independent data sets, data it hasn't seen during training, to make sure it's robust and generalizes well.
Test it on new examples.
Right. And crucially, this is where explainable AI or XAI techniques come into play.
Avoiding the black box.
Exactly. XAI methods help us understand why the model made a certain prediction, which features, potential biomarkers, were most important for its decision. This provides biological insights and builds trust in the model's output. It's not enough for the AI to just say this is a biomarker. We need to understand why it thinks so.
Makes sense. So what specific AI techniques are commonly used for biomarker prediction?
There's a range. In classic machine learning, ML, you see things like random forests and support vector machines, SVMs. They're good for classifying samples, and they can also rank features by importance, highlighting potential biomarkers. Gradient boosting machines, GBMs, are also popular, especially for handling nonlinear relationships.
Okay, standard ML. What about deep learning?
Deep learning, DL, is increasingly used too. CNNs might analyze spatial data, like patterns in tissue scans related to certain biomarkers. Recurrent neural networks, RNNs, can model time series data, how biomarker levels change over time, maybe in response to treatment. And autoencoders can help reduce the high dimensionality of omics data to identify latent features that might represent underlying biological processes linked to biomarkers.
And unsupervised methods for discovery.
Right. Unsupervised learning techniques like clustering algorithms, k-means, DBS can group samples with similar biomarker profiles, potentially discovering novel disease subtypes. Principal component analysis, PCA, is often used for dimensionality reduction and visualization, helping researchers see patterns in the data.
And you mentioned integrating different data types.
Yes. Multi-omics integration is a powerful approach. AI methods are used to combine data from multiple layers, genomics, proteomics, metabolomics, etc., to identify biomarkers that reflect complex interactions across different biological processes. This often gives a more robust and comprehensive picture of the disease state.
It sounds like these techniques are already being applied.
Oh, absolutely. AI has successfully predicted or helped identify biomarkers in many fields. In cancer, for instance, analyzing gene expression and proteomics data for early detection markers, like improving on existing ones or finding new ones, and also predicting which patients will respond best to certain therapies.
What about other diseases?
In neurodegenerative diseases, AI is used to analyze imaging data like PET scans combined with proteomic or genetic data to predict biomarkers for Alzheimer's disease, like amyloid beta or tau protein levels. For infectious diseases during COVID-19, for example, AI helped identify immune response-related biomarkers for rapid diagnosis and predicting disease severity. And in cardiovascular disorders, predicting known biomarkers like troponins for heart attack or finding new inflammatory markers for monitoring heart disease risk.
So it's happening across the board. Are there specific software tools people use for this?
Yeah, there's a mix. Researchers use general-purpose AI libraries like TensorFlow, PyTorch, Scikit-learn, XGBoost, Keras. These provide the building blocks for creating the models.
The standard AI toolkits.
Right. Then there are more specialized bioinformatics tools, things like DeepChem for drug discovery and cheminformatics, Bioconductor for analyzing genomic data in R, ProteoWizard and MaxQuant for handling mass spectrometry data, String for PPIs, MetaMap R for mapping text to biomedical concepts. The list goes on.
And for the explainability part, the XAI.
Yes, specific tools for that are becoming essential. SAP, that's Shapley Additive Explanations, and LIME Local Interpretable Model Agnostic Explanations are two popular ones. They help researchers understand which features drove a specific prediction, making the AI's reasoning more transparent and interpretable, which is vital for clinical translation.
Okay, that's a great overview of biomarkers. Let's broaden our view again now. We've touched on genomics, proteomics, diagnostics, biomarkers. What are some of the other big areas across the life sciences where AI is making waves?
Oh, there are many. Let's talk about synthetic biology. This is essentially engineering life or biological systems for useful purposes, and AI is bringing incredible precision to it.
Engineering life. Sounds futuristic.
Yeah.
How does AI help?
Take CRISPR-based gene editing, for example. CRISPR-Cas9 is a powerful tool, but one challenge is ensuring it edits only the intended target in the genome. AI is significantly enhancing this. By analyzing genomic data to predict potential off-target effects, those unintended DNA modifications elsewhere in the genome. AI helps identify the optimal target sites that minimize off-target risk and also helps optimize the design of the guide RNA, the molecule that directs CRISPR to the right spot. This makes gene editing safer and more efficient.
Okay, making CRISPR better. What else?
Gene sequence optimization. If you want to insert a gene into an organism to make it produce something, say a drug or a biofuel, the exact DNA sequence matters. Machine learning and deep learning models analyze genetic data to determine the ideal gene sequence for a specific function, maybe to maximize protein production or stability.
Designing the code for optimal performance.
Exactly. Companies are using AI for this, and related to that is protein design and engineering. We talked about AlphaFold predicting existing structures, but AI is also used to design novel proteins from scratch.
Creating proteins that don't exist in nature.
Potentially, yes. Forecasting the structure and function from sequence allows scientists to computationally design new enzymes with enhanced activity, new therapeutic proteins, or even new biomaterials with specific properties.
Amazing. And what about engineering whole pathways?
Right, metabolic pathway engineering. This is about modifying the network of chemical reactions inside a cell to make it produce a specific compound more efficiently, biofuels, pharmaceuticals, industrial chemicals. AI helps pinpoint bottlenecks in existing pathways or design entirely new pathways by analyzing the complex interplay between genes, enzymes, and metabolites.
Optimizing the cell's internal factory.
That's a good analogy. And finally, in Synbio, AI is even helping with automated experiment design. It can help optimize the whole experimental process, predicting which experiments are most likely to yield valuable results, guiding researchers, and sometimes even controlling robotic lab automation systems.
Making the research itself more efficient.
Less trial and error, faster path to results.
Okay, synthetic biology is fascinating. What about drug discovery? You mentioned it earlier, but let's dive deeper. You said it's long and expensive.
Extremely. Traditional drug development is a really rigorous process, no doubt, but it's incredibly long and expensive. We're talking 5–10 years, sometimes longer, and costs can run up to or even exceed $2.5 billion per drug.
Billion with a B, wow.
Yeah. And the success rates are sobering. Only about 13.8%, maybe even less, of drugs that enter phase 1 clinical trials eventually get final FDA approval. It's a high-risk, high-cost endeavor.
So the need for improvement is massive. How does AI help specifically?
Several ways. One big one is virtual screening. AI methods can rapidly sift through enormous digital libraries of chemical compounds. Databases like Tumble or ZNC might have millions or billions of molecules.
Looking for potential drug candidates.
Exactly. AI models predict which molecules are most likely to bind to a specific disease target, like a protein, and have drug-like properties. This drastically narrows down the number of compounds that need to be synthesized and tested experimentally, saving huge amounts of time and money up front.
Screening existing chemicals. What about designing new ones?
That's de novo drug design. This is where generative AI really shines. Instead of just screening existing molecules, AI models are used to generate entirely new chemical structures from scratch, designing them computationally to have specific desired properties like high potency, good absorption, low toxicity.
Building the ideal drug molecule digitally first.
That's the idea, opening up completely novel chemical space. Another powerful application is drug repurposing.
Finding new uses for old drugs.
Exactly. AI analyzes vast amounts of data, drug properties, disease mechanisms, clinical trial results, scientific literature to identify existing drugs that might be effective against different diseases than the ones they were originally approved for.
We saw that with COVID, right?
Yes, that was a prominent example. AI played a role early in the pandemic, predicting that certain existing drugs, like dexamethasone, might have potential against COVID-19 based on analyzing biological pathways and drug interaction data. This approach also leverages real-world data, electronic health records, insurance claims to find drug disease associations that might not be obvious just from lab studies.
And AI helps, even after a drug candidate is found, in clinical trials.
Increasingly, yes. AI is playing a role in clinical trials, CTs. Models are being developed to assess the likelihood of a drug succeeding as it moves through the different phases of clinical trials, phase 1, 2, 3. Some algorithms are showing pretty good accuracy, around 80% in predicting phase transitions.
Predicting success or failure earlier.
Potentially, yes. This could help optimize trial designs, maybe identify patient subgroups most likely to benefit, and predict patient outcomes, ultimately aiming to make trials more efficient and increase the overall success rate of drug development.
Okay, huge potential in medicine. What about outside the lab, like ecology and environmental sciences?
Yes, AI is making inroads there too. Ecologists deal with massive data sets, often from remote sensing, camera traps, acoustic monitoring. AI helps them analyze this vast amounts of data.
For that kinds of tasks.
Monitoring and prediction. AI helps monitor changes in species populations and ecosystems, measure things like air quality, track how species distribution is changing, perhaps due to climate change. It's also used to build predictive models for complex events like wildfires.
Predicting where fires might spread.
Exactly. Or even identifying which animal species are likely carriers of certain diseases, like predicting which bat species might harbor filoviruses like Ebola. That's important for understanding zoonotic disease spillover risk.
That sounds crucial. What about data quality in ecology? Is that an issue?
It definitely can be. Think about citizen science platforms like iNaturalist, where people upload photos of plants and animals. It's amazing data, but sometimes there are errors, mislabeled species. AI can be trained to help clean this data, identifying potentially erroneous records.
AI checking the quality control.
Right. And AI also helps address challenges in data interoperability, making data from different national monitoring programs compatible so you can build global data sets. This is feeding into initiatives like the UN Environment Programme's Global Environmental Data Strategy, aiming for better data sharing by 2025.
And for conservation directly.
Yes, conservation applications, AI assists in tracking at-risk populations. You mentioned the MIT Elephant Book platform using computer vision to identify individual elephants in Kenya from photos. That's a great example. It allows monitoring without physically capturing and tagging animals.
But there are risks.
Yes, data sensitivity is huge. If AI helps map the precise locations of endangered species, that data needs extremely careful handling. In the wrong hands, it could actually facilitate poaching or habitat destruction. So ethical data management is paramount.
Absolutely. Okay, one more sector, agriculture and industrial biotechnology. Optimizing things for sustainability.
Exactly. In agriculture, often called precision farming, AI is making a big difference. AI-based machine vision can analyze images of plants much faster than humans, helping breeders identify and select improved crop varieties more quickly.
Speeding up crop breeding.
Yes, and drone technology equipped with AI is used to monitor crop health across large fields. For example, using advanced neural networks like EfficientNet V2 to detect plant diseases from drone imagery with incredibly high accuracy, like 99.99% in some studies.
Wow. Spotting disease outbreaks early.
Right. AI algorithms, often neural networks or genetic algorithms, are also used to optimize crop yield prediction for major crops like soybeans and maize, helping farmers make better decisions. And they inform smart irrigation systems, optimizing water use. AI-based smart sprayers can even identify weeds and target them specifically with herbicides, reducing overall chemical use.
Making farming smarter and greener. What about industrial biotech?
Right. Industrial biotechnology, sometimes called white biotechnology. This is about using biological systems, microbes, enzymes for sustainable manufacturing. AI and machine learning help optimize these bioprocesses.
Like making enzymes or biofuels.
Exactly. Optimizing conditions for enzyme production by microbes or maximizing biofuel yield from renewable sources like agricultural waste. Integrated AI models, combining neural networks and genetic algorithms, are used for this. AI also aids in metabolic engineering to create high-performing microbial strains, like engineering cyanobacteria to produce more biomass for biofuels.
Fine-tuning microbes for industrial jobs.
Pretty much. And AI even helps with things like real-time forecasting of market demand for bio-based products, or even quality assessment, for instance, using AI models trained on aroma profiles to assess the quality of beer during production. It's all about using biology more efficiently and sustainably for industrial purposes.
Okay, that covers a huge range of applications. This brings us back to something you highlighted earlier, the power of these foundation models across biomedical research. You said they're having a profound impact.
They really are. It's worth revisiting them specifically because they represent such a significant trend. What makes them so impactful, again, is that they're trained on these massive, diverse, often unlabeled data sets. They learn fundamental patterns in relationships in a self-supervised way.
Learning the language of biology, in a sense.
That's a great analogy. They learn deep representations of biological data, genes, proteins, cells, making predictions even for inputs or contexts they haven't explicitly seen before, much like a human expert develops intuition.
And you said their key strength is integration.
Absolutely. Their profound ability to integrate diverse data types, genes, amino acids, proteins, chromatin structure, single-cell data, even medical images and text, all within potentially a unified framework. This is huge. It helps researchers see novel connections between different biological scales or data modalities.
Pointing towards new hypotheses.
Exactly. Providing direction for unresolved questions, forming the basis for entirely new hypotheses. And as we mentioned, the field is exploding. Over 150 papers just in 2024, including preprints. It's a paradigm shift.
Okay, let's get concrete. Can you give some examples of these foundation models across different areas of Oni's and clinical practice? Maybe starting with transcriptomics gene expression.
Sure. In transcriptomics, a good example is Geniformer. It uses transfer learning. It was pre-trained on a massive data set of 95 million single-cell transcriptomes.
95 million cells.
Yeah, huge scale. And it can then predict tissue-specific gene network dynamics from single-cell RNA sequencing data. This is especially valuable where data is limited, like in rare diseases or for tissues that are hard to sample.
What else in single-cell analysis?
There's SCGPT, the single-cell GPT we mentioned, introduced in 2024, pre-trained on about 30 million cells. It's a generative tool designed to distill insights from single-cell data annotating cell types, inferring gene networks, clustering cells, even integrating multi-omic data sets within the single-cell context.
So understanding cell diversity and function better?
Right. And another important one is SCVI, single-cell variational inference. It uses deep learning and statistical methods to model the distributions of gene expression across large single-cell datasets, enabling tasks like visualization, clustering, and finding differentially expressed genes between cell populations.
Okay, transcriptomics covered. What about genomics, the DNA sequence itself?
We touched on some earlier. DeepSea, which came out back in 2015, and the more recent Informer from 2021. Both use deep learning to predict the effects of non-coding genomic variants on things like chromatin accessibility and epigenetic regulation.
And Informer handles those long-range interactions.
Exactly. It's specifically optimized to include interactions up to 100,000 base pairs away, which we now know are critical for accurate predictions of variant effects on gene expression. Then there's DMERT, adapting the BERT language model to contextually understand DNA sequences and predict important regulatory regions.
Understanding the grammar of the genome. What about structural biology?
Well, the elephant in the room is AlphaFold. We've discussed its impact on predicting 3D protein structures with near experimental accuracy. The latest version, AlphaFold 3, is pushing further, focusing specifically on predicting the structures of complex biomolecular interactions, how proteins bind to DNA, RNA, or other proteins. This work even contributed to a Nobel Prize.
Truly foundational. Okay, you also mentioned spatial biology, understanding cells in their tissue context. Are there foundation models there too?
Yes, this is a rapidly emerging area. Niche-Former is one example. It's trained on both dissociated single-cell data, where spatial context is lost, and spatially resolved transcriptomics data.
Bridging the gap. Yeah.
Exactly. It aims to predict the spatial microenvironment context for dissociated cells, helping us understand how a cell's neighborhood influences its behavior. Another one is Nove, developed in 2024. It uses graph-based learning, pre-trained on about 30 million cells across 18 tissue types.
What does Nove do?
It enables comparison of spatial domains, distinct tissue regions or niches across different samples and experiments. Importantly, correcting for technical batch effects that often plague these comparisons. This allows for more robust analysis of how tissue organization changes in disease or development.
Okay, spatial context is key. What about understanding the language about biology, the literature?
Right, biomedical language. Here we see models like BioBERT and BioElectra. These essentially take powerful general-purpose language models, like BERT or Electra, and adapt them using transfer learning on vast amounts of biomedical text, like PubMed abstracts.
Making them experts in medical jargon.
Pretty much. This significantly improves their performance on specific biomedical text mining tasks, extracting relationships, answering clinical questions, summarizing research compared to the general-purpose models.
And applying these language and vision concepts in actual clinical practice.
Yes. We mentioned CICO and CH, contrastive learning from captions for histopathology. It's trained on over 1 million image caption pairs from pathology reports. By combining visual learning from the images with language understanding from the captions, it shows potential to outperform visual-only models in analyzing tissue slides, classifying disease, generating captions, segmenting regions of interest.
Combining modalities for better clinical insights.
Exactly. And another example is TXGNN. It's a neural network trained on a large medical knowledge graph. Its goal is drug repurposing, identifying potential new uses for existing drugs by analyzing the relationships in the knowledge graph. Its predictions apparently align well with current off-label drug use practices, suggesting it's capturing real clinical patterns.
So these foundation models are being applied to specific diseases. Can you give examples of the impact?
Absolutely. The real test is whether they lead to new biological insights or clinical advancements. And they are starting to.
Like in cancer.
Yes, in cancer. For example, SCGPT was used in breast cancer mouse models. It helped pinpoint factors driving resistance to therapy, identifying a specific gene, C5RR1. Inhibiting this gene actually resensitized the tumors to the treatment in the lab.
Wow, a direct therapeutic hypothesis.
Right. Another example. SCVI modeling identified changes in the tumor microenvironment, the surrounding stromal cells that influence prostate cancer progression. They derived a transcriptional signature from this that could predict which patients were more likely to develop local metastasis.
Okay, cancer. What about neurodegeneration?
In neurodegeneration, SCVI was used in a mouse model of multiple sclerosis. It identified a specific subset of T cells that seemed to be driving the expansion of B cells, which are involved in the disease pathology. In Parkinson's disease, GeneInCell, analyzing patient data, predicted a key role for a transcription factor called NFXC2 in regulating neuroinflammation. This prediction was later validated experimentally in a mouse model.
Finding molecular drivers of disease. Any examples in infectious disease?
Yes, for infectious disease. AlphaFold predicted the structure and potential inhibition mechanisms for EPA, which is a multi-drug efflux pump in Mycobacterium tuberculosis. These pumps actively kick antibiotics out of the bacterial cell, contributing to drug resistance. Understanding its structure is a big step towards designing drugs to block it, potentially helping treat drug-resistant TB.
Tackling antibiotic resistance. What about regenerative medicine?
Stem cells and regenerative medicine. AlphaFold's structure prediction helped researchers understand how a protein called MYCT1 modulates the self-renewal and engraftment ability of human hematopoietic stem cells, HSCs, the cells that produce all our blood cells. They figured out it localizes to endosomal membranes, giving clues about its function. This kind of basic science insight is crucial for developing better stem cell therapies.
Understanding the fundamentals. Rare diseases.
Rare disease. DeepSea, that model for non-coding variants, was used to investigate ultra-conserved non-coding elements, UCNEs. These are stretches of DNA outside of genes that are highly conserved across evolution, suggesting important functions. DeepSea helped reveal their roles in retinal development and linked some to rare eye diseases, helping researchers narrow down the list of candidate variants to investigate clinically in patients.
Helping find the genetic cause when it's really hard. Cardiovascular disease.
Cardiovascular disease. SCVI modeling analyzed heart tissue from atrial fibrillation patients at the single-cell level. It identified specific transcriptional changes happening in different cell types. For instance, it highlighted a gene called ATRNL1 as a key player in how heart muscle cells, cardiomyocytes, respond to stress. It also found unexpected expression of another AFib candidate gene, KCNN3, in lymphatic endothelial cells, suggesting a previously unknown role for these cells in the disease.
Uncovering unexpected biology. One more gene therapy. Getting treatments into cells.
Right, gene therapy drug delivery. This is a huge challenge. AlphaFold modeling, combined with other techniques, helped identify interactions between engineered adeno-associated virus, AAV, capsids, the protein shells used to deliver gene therapies and proteins in the human body. Specifically, proteins involved in immune response, which can limit therapy effectiveness, and proteins involved in crossing the blood-brain barrier, BBB, like LRP6.
So understanding how to design better delivery vehicles.
Exactly. This work informs how to engineer AAV vectors to better evade the immune system or to more effectively cross the BBB for non-invasive delivery of therapeutics to the brain, which is a major goal for treating many neurological disorders.
These examples really illustrate the power.
Mm-hmm.
But you mentioned earlier, these models require serious computing power.
They absolutely do. That's a practical constraint. While these foundation models are incredibly powerful, they require significant computational resources to train and often even just to run efficiently. We're talking serious GPU power.
GPUs, graphics processing units?
Yes. Training a large foundation model is incredibly computationally intensive. It can require processing many terabytes of data and relies heavily on GPU acceleration. Remember Meta's ESM fold? That used 2,000 GPUs. So this necessitates advanced computing infrastructure and specialized software tools, which represents a significant barrier to entry for smaller labs or institutions that don't have access to that kind of hardware.
Okay, so computational demands are a hurdle. This leads us naturally into the broader conversation about navigating the future, the really significant challenges and profound ethical considerations that come with deploying AI in the life sciences.
Absolutely critical discussion. We can't just focus on the potential without acknowledging the pitfalls.
And maybe the place to start is that old adage, garbage in, garbage out. Data quality seems paramount.
It's absolutely fundamental. This is often called the garbage in, garbage out problem. AI models are, as the research states, extremely reliant on the data sets on which they are developed. If the input data is inconsistent, incomplete, or just plain wrong, the model's performance will be severely impaired. You can't expect good output from bad input.
And you mentioned earlier that biological data, like from proteomics, is inherently messy.
Exactly. Fields like proteomics generate data characterized by high data variability and noise. This poses continuous challenges for AI models trying to extract reliable biological signals. It's not always easy to tell what's real signal and what's just technical noise or random fluctuation.
What about outright errors in the data?
That's a big issue too. Even large, widely used public data sets, like those aggregated from citizen science platforms, can contain misrepresented or mislabeled data. We talked about AI being used to clean this data, but it highlights the underlying problem. The raw data often isn't perfect, and errors can propagate if you're not careful.
And this leads to the really thorny issue of bias, right?
Yes, bias in training data is a major ethical and practical concern. AI models don't just learn patterns. They can also learn and even exacerbate pre-existing discrepancies present in the data. They can reinforce societal biases against underrepresented groups if those groups are well represented or are represented inaccurately in the training data sets.
You gave that example earlier with facial analysis.
Right, the Deep Gestalt tool for facial dysmorphology. Its performance for diagnosing Down syndrome was initially much lower for individuals of African heritage, around 37%, compared to those of European origin, 80%. Retraining with more diverse data improved it dramatically, but it's a stark warning. And the research emphasizes that genetic disease susceptibility modeling is also predisposed to differential performance across demographic groups. We have to be incredibly vigilant about building and testing models on diverse representative data.
And beyond bias, what about just knowing where the data came from, data provenance?
That's another huge challenge. Tracking the origin, quality, and processing history of every single data point within and across massive interconnected databases is incredibly difficult. A particularly tricky aspect highlighted in the sources is that no computational tools are currently available that can reliably identify computationally generated data versus real experimental data.
So you might not even know if some data was faked or simulated.
Potentially. This makes manual validation almost impossible as data volumes explode, and it creates a real risk of errors propagating silently through the system if generated or poor quality data gets mixed in without clear labeling or tracking.
Okay, data quality and bias are huge.
Yeah.
What about understanding the models themselves, the black box dilemma?
Right. This is a persistent criticism, especially for complex deep learning models. They often suffer from black box characteristics. It's difficult, sometimes nearly impossible, to understand how the model arrived at a specific prediction.
You get an answer, but you don't know why.
Exactly. And this lack of transparency is a major barrier to trust and widespread adoption, particularly in high-stakes fields like clinical medicine or scientific research, where understanding the underlying mechanism or reasoning is often just as important as the prediction itself. It hinders our ability to derive biological insights from the model's workings.
Are there practical consequences to this lack of transparency?
Yes. One issue highlighted is non-determinism and model drift. Studies evaluating advanced LLMs like GPT-4 in clinical workflows, such as variant assessment, found that the model's output could vary even for the same input, and its performance or behavior could change over time as the model is updated. The researchers concluded this must be addressed before deployment in diagnostic settings. You need reliability and consistency in healthcare.
Unpredictable AI is not good for doctors or patients.
Absolutely not. And this ties into the risk of misplaced trust. Generative AI, LLMs in particular, can be very fluent and persuasive. They can sound highly confident even when they're wrong. In healthcare, a wrong answer can mean a misdiagnosis or worse.
The stakes are too high.
Exactly. So building robust risk management frameworks, ensuring rigorous validation, and always having human validation and oversight are crucial responsibilities. Predicted biomarkers, for example, need extensive experimental and clinical validation to ensure reliability before ever being used to guide patient care. We can't just take the AI's word for it.
Okay, data issues, trust issues. What about just the practicalities, computational resources, and implementation hurdles?
We touched on the demanding infrastructure. Training large foundation models is incredibly computationally intensive, needing serious hardware, especially GPUs. This requires advanced infrastructure and specialized expertise, which isn't available everywhere.
And the cost.
Cost barriers are real, not just for training huge models, but even for deploying certain AI applications. Think about those advanced drones with high-res sensors for precision agriculture. They can be highly expensive for small farmers, potentially widening the gap between large industrial farms and smaller operations.
So access isn't equal. What about integrating AI into existing systems?
That's another hurdle. Integration challenges persist, especially when trying to integrate multimodal data, genomic sequences, imaging reports, electronic health records into unified, seamless, and clinically robust pipelines. Getting different systems and data types to talk to each other effectively is often harder than building the AI model itself.
And the rules, the regulations.
Regulatory lag. This is a big one. As one source put it, if technology is racing ahead, regulation is struggling to keep pace. Laws regarding data privacy, data use, model validation, and accountability differ dramatically across countries and even regions.
Like using medical data.
Exactly. Rules about whether anonymized medical data can be sent overseas vary hugely. Some places lack comprehensive data privacy laws altogether. Consent forms for using patient data are a minefield. What happens if a patient consents to their data being used to train an AI, but later withdraws consent or asks for their data to be deleted? How do you track and remove that data's influence from a trained model? These are complex legal and technical questions without easy answers yet.
Wow. And finally, the people involved.
Mm.
The human factor.
Crucial. A significant challenge, as highlighted in our reading, is persuading people that you were there to augment, not replace them. Building trust among clinicians, researchers, lab technicians, and patients is essential. Without it, AI adoption will never scale, no matter how good the technology is.
People need to trust it and want to use it.
Right. And we also need robust support systems for when AI tools inevitably make mistakes or fail. There needs to be clear protocols for human oversight, intervention, and correction. It's about human-AI collaboration, not just handing things over to the machine.
Okay, these challenges really lead us into the final area. The profound ethical and societal implications. These go beyond just the technical hurdles.
Absolutely. We're talking about fundamental questions about privacy, fairness, accountability, and the very future we want to build with this technology.
Let's start with privacy and security. That seems like a major concern, especially with generative AI.
It is. Deep concerns exist about how personal identifying information, PII, is handled. When people interact with Gen AI systems, where does that data go? How is it stored? Who owns it? Could it be shared or sold? These questions are critical, especially when dealing with sensitive health information.
And not just health data, you mentioned ecology too.
Right. The use of camera traps, audio recorders, and ecological monitoring raises questions about inadvertently capturing data about people or communities without their explicit consent. Balancing conservation goals with privacy rights is an ethical tightrope walk.
Which points to the need for accountability and regulation.
Imperative. Establishing clear accountability frameworks. Who is responsible if an AI makes a harmful diagnostic error? And developing robust regulations is essential to prevent misuse and ensure responsible AI practices. Companies developing and deploying AI need to work closely with legal experts and policymakers to define these roles and responsibilities clearly.
And there's the scary potential for misuse.
Unfortunately, yes. There's a tangible concern that generative AI, particularly models trained on biological data, could potentially be misused, for example, to design new pathogens or enhance existing ones. Some researchers are already building safeguards, like explicitly excluding virus genomes from training data for certain generative models, but it's a risk that requires ongoing vigilance and discussion.
That's sobering. What about fairness, access, and equity?
This is a double-edged sword. AI has the potential to improve access to expertise or diagnostics in underserved areas. But as we discussed with cost barriers and potential biases, if not managed carefully, AI may also exacerbate pre-existing discrepancies. Ensuring equitable access to the benefits of AI and mitigating potential harms for vulnerable populations has to be a central goal.
This sounds like it needs global cooperation.
It really does. Highlighted in the sources are initiatives like the UN's proposed Global Digital Compact. The aim is to foster international collaboration to narrow the gap between rapid technological evolution and the development of global governance frameworks, working towards an open, free, and secure digital future for all. We need shared principles and standards.
And what about the people working in the field? Expertise and education.
There's a clear and growing need for improved educational programs for biologists, clinicians, and life scientists, not just on how to use AI tools, but also emphasizing best practices in data engineering, understanding model limitations, interpreting results critically, and navigating the ethical considerations. We need a workforce that is both AI literate and ethically aware.
So at the end of the day, AI is a tool.
Exactly. A powerful tool, but still a tool. It's crucial to acknowledge, as one review put it, that AI-based approaches will not always lead to improved categorization or better prediction than present methods. It's not a magic bullet. AI should be employed within the proper context to address a pertinent question or resolve a significant issue, always with human judgment, critical evaluation, and ethical oversight.
It's a really important perspective to maintain. Okay, as we wrap up this deep dive then, it feels like we've covered a massive amount of ground.
We definitely have.
You've really seen the incredible breadth and depth of AI's impact across the life sciences. I mean, from the incredibly intricate dance of genetic variants and protein structures, all the way to optimizing the global food supply and helping us understand complex ecosystems.
Yeah, the scope is just immense. AI is clearly accelerating discoveries at an unprecedented rate. It's fostering a much deeper understanding of biological complexity, and it's truly transforming what we thought was possible to areas like medical diagnostics, drug development, and so much more.
It really feels like more than just a passing trend, doesn't it?
Yeah.
It seems like a pivotal shift in how we approach biological research and its applications.
I completely agree. It represents a fundamental change. And those foundation models we discussed in particular are proving to be incredibly powerful integrators of diverse data. They're paving the way for insights we couldn't have imagined even a few years ago, allowing us to ask entirely new kinds of questions.
And it's a significant but. We've also spent a good amount of time shining a light on the critical challenges.
As we must.
The absolute necessity for impeccable data quality, the ongoing quest to make these powerful black box models more interpretable and trustworthy, the huge ethical imperatives around privacy, mitigating bias, and ensuring responsible development. And, of course, the urgent need for robust, thoughtful regulation that keeps pace with the technology.
Yeah, this journey is definitely ongoing. And what comes through clearly from all the sources is that the human element, your understanding, your critical thinking, your ethical judgment, remains absolutely central to navigating this journey successfully. AI is a tool. Humans wield it.
That's a great way to put it. So maybe a final provocative thought for you, our listener, to reflect on as you go forth from this deep dive.
Okay.
Consider this. AI isn't just changing what we can discover in biology. It's fundamentally changing how we interact with, and maybe even engineer, life itself.
That's a profound point.
So what new ethical responsibilities does this profound power bring? How can we collectively ensure that this incredible technology is harnessed wisely, responsibly, and truly for the benefit of all humanity, while also respecting the delicate balance of the natural world we depend on?
Big questions, no easy answers.
No easy answers indeed. But the future of the life sciences powered by AI is unfolding right now, all around us. And hopefully after this discussion, you're now better equipped to understand it, engage with it, and maybe even help shape it.